Good afternoon. Hope everyone is enjoying their lunch okay. I'll get mine afterwards, after my nerves have settled a little. I think I should start off by introducing who antibody analytics are. We are a collaborative CRO based in Scotland. My accent probably gives that away a little bit. We are just outside of Glasgow. We're about 20 miles outside of Glasgow, in a place called Motherwell, and we have our own purpose-built labs there. We have been growing steadily over the past 5 years, mostly, since COVID. And we now employ over 100 employees, and most of these guys are experts in immunology and bioanalytics. We offer primary cell assays, building in bespoke molecular biology models, and a lot of our customers, we have a very global customer base. The modalities that we look at are primarily antibodies, given the name, but we also do a lot of small molecule, and we now do cell therapy modalities as well. I think it would be fair for me to assume that everybody here has a deep appreciation for how complex the tumour microenvironment can be. And at antibody analytics, we try and recapitulate a lot of the cell types and processes that happen in that tumour microenvironment that contribute to disease progression. But for the purposes of today, I would just like to quickly cover what we can offer in terms of T-cell hyperresponsiveness assays. And another theme that I've seen coming up quite a lot in this meeting as well is the issues around target antigen density. And then I'd like to, if I've got time, come back and just briefly talk about some of the other things that we're starting to develop in terms of looking at the tumour microenvironment as a whole and being a bit more relatable to clinic. So T-cell hyperresponsiveness, there are different ways in which you can have hyperresponsiveness in T-cells. You can have active regulation, which we have assays that can help look at that as well by Tregs or macrophages. But we have had a lot of interest in T-cell exhaustion. So we're talking about that chronic antigenic stimulation that results in a functional state of hyperresponsiveness, where the T-cells no longer proliferate in response to stimuli, and you have loss of their effective function. So they don't kill anymore or produce cytokines in the same way when you compare to a conventional non-exhausted counterpart. So for some time now, we have had a polyclonal-based system where we repeatedly stimulate primary human T-cells, CD4s and CD8, to produce an exhausted cell phenotype. So these cells typically have high levels of LAG3, express high levels of TOX, PD1, and phenotypically they look like exhausted T-cells. If you take these cells and put them in an MLR, a secondary functional assay, we see that compared to a single stimulation primary T-cell, these T-cells don't proliferate, they fail to produce levels of cytokines, and equally, what's important is we can actually partially recover that with checkpoint inhibition. In this case, this is showing data for using nivolumab. Normally, we set up these assays to look at can you reverse T-cell exhaustion, but what has been a clear theme throughout the meeting of the last few days is that it's also relevant to not just checkpoint inhibition and reinvigoration, it's about how your therapy may actually drive T-cell exhaustion as part of that process. So if you have T-cell engagers, what happens when you repeatedly stimulate T-cells with your modality? And also for things like CAR-Ts, if you have a high affinity interaction, repeated antigenic interaction, do you drive exhaustion in these systems as well? So this is something a lot of our customers are now coming to us to do, and we are now offering assays where we can do these multiple activations in these systems to drive exhausted cell phenotypes. So that's all based on our initial first generation exhaustion assay, but one of the key things that's important to the function of natural T cells is antigen specificity. So we've been currently developing or expanding our antigen-specific T-cell assay repertoire. Currently, we can do recall for things like CMV or use various peptides and pre-screen donors to demonstrate T-cell functionality. But that's using model antigens. In terms of looking at the IO market, we wanted to develop something or utilize systems that were more directly relevant. So what we have been doing is we are now geared up to, To start out with small populations of antigen-specific T cells. In this instance, this is healthy HLA-A2 donors, where you already have a slightly elevated frequency of MART1 specific T cells in there. And we can stimulate these with specific antigen and expand the cytokines over a period of 15 days to enrich that population and give us large banks of mixed T cells. What we're then able to do is magnetically enrich these with tetramers to give high purity antigen-specific tumour antigen relevant T cell populations. And these are nicely functional. We can use these in a target cell cytolysis. We can incubate them with MART1 endogenously expressing target cells such as MeWOs. And we can use them in our intelligence-based assay, where we use impedance to measure real-time cytolysis over time. And here we can see as you increase the number of antigen-specific T cells in the system, you start to get increased cytolysis. So this is just where we're at just now, just to give you a flavour of what we're doing with antigen-specific systems. The aim is to offer this as a service where you can use antigen-specific T cells to test your drug modality, but equally, we want to then progress to developing our second-generation antigen T cell exhaustion model, where we'll be using antigen-specific T cells in that capacity. So this is just to give you a flavour of some of the things we can do. But the second theme, and this is where I'm going to focus on most of the talk that's come out, is this issue of target antigen density. What type of response do you get with your therapy? So why does target antigen density matter? Well, it matters for multiple immune therapies because where in an ideal situation, you'll have a high level of tumour antigen expression and you get good therapeutic effect. But we all know that the target antigen expression is very heterogeneous, and you have situations where you have low expression, and then you also have low antigen expression on healthy tissues as well. And this makes it really difficult in terms of targeting how effective your therapy should be. So this has become a real clear preclinical and clinical consideration. And this is just to put up an example here from the FDA, where they are highlighting these undesired targeting of healthy and normal tissues that also induce the intended target antigen and a non-target of tumor consequence. So the conventional approach to assess the impact of antigen density and the efficacy of your therapeutic is to use a panel of cancer cells. You select different target antigen cells based on their range of target antigen expression. And this is fine, but there are a number of limitations associated with this, okay? First of all, the logistics. Can you access all these different populations of cells? And there's a cost associated with that, particularly where there's a lot of licensing surrounding the acquisition of the cells. There are methodological limitations, i.e. the range of expression may be limited, or you might have big gaps in antigen expression. And then you have confounding factors by using target antigen cells from different backgrounds. So when you culture those cells with your immune effector cells, those cells may be more susceptible to factors independent of the antigen density because of the different cell backgrounds. So what would the ideal system look like? You would have a common genetic background, single cell. That ensures that absence of confounding factors from the heterogeneity of a panel of different cell populations. You should have a large dynamic range of expression. Okay, that would enable assessment of antigen abundance across a wide range of expression levels, from hundreds to hundreds of thousands of target antigen receptors. That should give you a more comprehensive analysis, and that should be finely titratable. That gives you a good control over the antigen expression, and that's crucial for helping you understand the impact of varying densities on your actual therapeutic efficacy. Availability of a negative control is always helpful, so you can have your target cell population there that doesn't express the target antigen. And ideally, all of this should be compatible with immune cell bioassays. So over the last couple of years, we've been producing a solution for this, and we've called this Index 2. It's inducible expression 2. It's not the second generation. Don't feel you missed out on Index 1. Index 2 is because we can induce 2 target antigens with this system. The way we've got this set up is that you can have inducible target antigen expression by increasing the dose of a small molecule inducer. And then we can put these range of antigen expressing target cells in our functional assays to look at cytolysis, flow cytometry to look at production of activation markers on T cells or NKs, whichever system you want to use. And then we can actually threshold where you have, we can do threshold determination to identify when your therapeutic is becoming most active. So how have we been doing that? So Index 2, it's a chemical induced proximity based system that gives us inducible expression. In essence, we have a small molecule that will allow dimerization of these 2 protein partners. And by combining these partners with a DNA binding domain and a transactivation domain, in the presence of the small molecule inhibitor, you basically get dimerization and create a molecular switch. So this allows us to have increasing expression of a gene of interest when you start to increase more of the small molecule and get more of these dimerizations. There have been a few SIP systems out there already using small molecule proteins to induce the dimerization. We have tacrolimus-based ones and gibberellin, but the 2 that we have focused in on are abscisic acid and indole 3-acetic acid, so ABA and IAA. And these are plant auxins, they're plant hormones. And the advantage of those is that they don't impact your human immune system when we use them in our cellular bioassays. So this is an example of some of the off-the-shelf systems that we have been setting up. We usually go for a CHOK1 background. We can do this in whatever cell line that you would like, which is most applicable. But we've been going for the CHOK1 just because it's easy for us to work with. It's readily available, and it transduces nicely. We make these. We have a lot of single antigen expressing cell lines for off the shelf, targeting the HER2, EpCAM, CD19, BCMA, and you can see we always get this really nice dose induction of expression with increasing concentrations of the small molecule inducers. And relatively, well, this data is from last year, the end of last year. We set up our first dual expressor system as well, so we can have IA induced and ABE induced driving 2 different target antigens and varying the levels of expression of those within the same target cell population. So if I can talk to you a little bit about a case study, particularly in the context of CD19, and the key thing here is the consideration of immune escape, okay? As you're aware, there are several approved CD19 targeting therapeutics out there, and although that they're very successful, a proportion of patients that are treated with them don't respond or relapse. One of the reasons that this seems to happen is linked to the levels of CD9 expression in these patients. And a couple of examples here that relate to the different expression of CD19 and escape mechanisms. But there was a paper by Spiegel and colleagues that correlated the levels of CD19 expression to responses with the CAR-T/CAR treatment. And in that paper, what they did was they used quantitative flow cytometry to define the levels at which CD19 expression might predict escape or effective treatment. And this was found out and identified to be about 3,000 receptors per cell. So you can imagine a situation for CD19 where you have anywhere above that 3,000 receptors, you have a good response, but below that you may have resistance. So ideally, if you wanted to avoid the escape, then your molecule should be able to function across that range of expression levels. So this is some work that we did with Clown Therapeutics, and they've kindly allowed us to share this for the day. This is using their CLN-978. It's a highly potent CD19 targeting T-cell engager. So they have used it specifically for treatment of B-cell malignancies. It has an HSA binding domain to extend its half-life, and through a number of assays that we've done in-house ourselves with them, we can show that it's very potent. It drives cytotoxicity. In Ramos and Raji cells, we have good levels of T-cell activation in a dose-dependent manner, and that's associated with increased levels of interferon gamma and TNF by the T-cells in that assay. So what happens if we use this in our receptor activation thresholding type assay? So we have CHO-K1 CD inducible CD19s. We incubate those with primary isolated T cells, isolated from our cryopreserved on-site donors. And then we did a comparison between the Clinton molecule and I would like to say blinatumomab. However, I find that really hard, so I'm going to call it Blincyto for the rest of this, if that's all right. And then at the end of the assay, we do cytokine analysis, and we have real-time cell cytolysis throughout that as well. And I think that this data really clearly demonstrates the difference in the two molecules. The Clinton molecule, we can see here right from the get-go, at the low levels of CD19 receptor expression, you get much higher levels of cytotoxicity using their system compared to the Blincyto. And when we look at the when we look at the interferon gamma response, we can always get a much stronger response with the Clinton molecule versus our benchmark material. So then that lets us overlay the readouts in terms of a threshold. So our benchmark material, we see, works very well, down to about 843 receptors per cell in terms of cytolysis. So that should be below the kind of low probability, or starting to get in that high probability of resistance. So that's okay, that's quite good. The interferon gamma response, you need a lot more receptors per cell to drive that other effector function. Whereas in comparison to the Clinton molecule, we see cytolysis is low down as 81 receptors per cell, and that interferon gamma response is very effective at that level as well. So this molecule is very potent. So in an ideal world, if you're worried about the high probability of resistance, then we would expect this molecule to hopefully be a bit more effective. But what other approaches are people taking to look at immune evasion? You can target that one antigen, or you can make a modality that targets more than one tumor associated antigen in your system. And we see a lot more of that these days. So to minimize that risk of relapse or refractory disease, then we target multiple antigens. And this is just some data that we literally generated last week using our new dual inducible system. We made our own in-house CAR T, targeting CD19 and 22. And we have our inducible CHO-K1s, where we have CD22 and 19 driven by the ABA or the IA separately. Put these guys together with the CAR T in a dish, leave them for 3 days, and we measure the target cell cytolysis. And we can see with increasing doses, so along the bottom here is where you have the CD19 on its own, but increasing concentrations, then you have increasing cytolysis. And on this axis, you get the same with the CD22. The more target antigen you have, the more cytolysis you've got. But we find that the hottest area that we have in the heat map, we have this accumulative is not the word I'm looking for, additive effect when we have both antigens expressed at the high level. And this is more clearly marked again with the interferon and gamma receptor. So this is just a proof of concept, if you like, that we're now set up and ready to do these types of assays and do threshold determination where you have more than one target antigen in your system. So the index 2 system, in summary, it allows the estimation of antigen threshold for initiation of your effective functions. And it's important to note that the threshold may be different for different effective functions, and that's what's really good about this system. You can have a complex bioassay at the end and tailor your molecule to give the exact profile that is most useful for your needs. allows us to do hazard assessment as well. So I'll come to talk a little just after this about how we can access primary disease state material, but we can therefore identify the healthy tissue expression levels and the disease tissue level expressions of your target, and then that lets us start to model the on-target off-tumour side effects that you may be worried about in an in vivo context. I've shown you data there using TCEs and CAR-T, but it's equally applicable for using monoclonal antibodies in an ADCC, and also for antibody-drug conjugates as well. And we've developed a number of off-the-shelf cell lines that you can just come to us and use straight away in your assays. But if you have complex targets, talk to us. We do on-demand cell line development, and we will build a bespoke cell line for you to use as well. So just in the last couple of minutes, I'd just like to give you a flavour for what we're thinking about in terms of the tumour microenvironment as a whole, and increasing the complexity of the systems that we can offer. As immunotherapies evolve, there's an increased demand for a more physiologically relevant assay, and we've seen some really nice talks over the last couple of days of people using organoids, tumoroids in their systems. And these take on these kind of explant co-culture systems and 3D assays. So we're currently starting to develop our own approaches, and it's very preliminary. It's just to give you a flavour. But we are now using patient biopsy material to develop these types of co-culture systems to hopefully develop 3D assays. So back home in the UK, we have our NHS, which gives us very detailed information about patient care. And we have set up a collaboration with what's called the Glasgow Biorepository. So all the main surgeries are done in this very localised place in Scotland across the Central Belt, and that allows us access to primary biopsy material, excised material from operations, not just for IO, but for autoimmunity as well. And because we're so local, we get the tissue straight from clinic, and it's in our lab within 2 hours. So it's pretty much as fresh can be when we get it. Yeah, that's basically what was covered. So the basic setup that we have, these are just some examples of the tissues that we've had recently. We get breast and ovary. We get a lot of colorectal carcinoma. We can get that quite frequently, and we've also had some pancreatic cancer samples as well. A nice man goes over to collect it from the biorepository in a taxi and brings it back to us, gives it in the lab, and then we can process that sample, either getting to the stage where you can make small organoids, like cut the tissue up for use as a 3D structure, or we can fully process it and generate single cell suspensions to use in different types of assays. So what have we been doing with those recently? This is just to show an example of a colon sample that we've had, and it just basically demonstrates the fact that we can make single cell suspensions. We have got lots of lovely, nice, viable cells. We can phenotype these, identify the different infiltrating lymphocyte populations. We can take this whole suspension and do functional assays with them straight off the bat. So this is just where we took this whole sample with the target cells in it, and we gave a variety of stimuli. So we gave a direct signal 1 through the TCR in the presence of nivolumab, signal 1 with some IL-15 on its own, and the IL-15 with signal 1, we see interferon gamma secretion. We've got some flow data as well that shows it can drive proliferation in that context as well. And as might be expected from some of the clinical data, we don't really see any real effect with the nivolumab because adenocarcinoma has that association of being refractory to PD-1 checkpoint inhibition. And in the last couple of things we're working on just now, last week, before I got on the plane to come here, we banked our first TILs for use in both our 2D and 3D assays. We're currently growing up the stroma that came from this sample to have primary tumour cell lines with a view to building organoids and 2D structures. So hopefully sometime in the next 6 months, we'll be able to say to you, like, here's our lovely assay, everything's all together, and we can do these 3D cultures with you. So yes, that's where we're heading just now. So in conclusion, antibody analytics, we offer primary human-based in vitro assays, and we can use these effectively to evaluate your immune-targeted cancer therapeutic. Assay design is always key to success, right? We should always focus on a specific question, and we like to always have a collaborative discussion with you guys to find out what you really need. Most of the people in our team have come straight over from an academic background. They do get really excited about the science, myself included, and we will probably just be as excited as you are with the data that we get out of these assays. And immune and target, considering the right immune cell and target cells are key to how we design these assays, and we always take care to consider how and why they're selected. So what is the disease state? What time point are you looking to treat at? Because, you know, in vivo disease is complex, and we want to kind of model the scenario best we can. And just to finish up, I'd like to say that this is an IO meeting, but the flip side of a lot of the immune processes that we try to switch on in IO, we want to switch off in other pathologies such as autoimmune disease. So we do a lot of work in those situations as well, and we've recently started doing some allergy. So if you are working on a therapeutic, keep in mind that you may be able to repurpose it for another function someplace else. I'd just like to acknowledge funding that we've had from UK RI, Scottish Enterprise, particularly Cullinan for letting us share their data and being a long-standing partner with us. We've done some really nice work with them. And the NHS, particularly for the biorepository for the guys in Glasgow. Thank you very much for your time. I hope you've enjoyed your lunch.
All right, do we have any questions for our speaker?
So I really liked your differential induction of various molecules on the cell surface. Two questions. One is, have you confirmed it with antibodies and doing a titration to know that how many molecules you're actually upregulating?
Yeah.
And the question is how stable it is. It looked to me like you were using prolonged culture periods, co-culture periods. And the question is, once you've induced a change, does it last an hour, a day, 3 days, 7 days?
Yeah. So the quantification we do is it's flow cytometry based. So we stain the cells, and we use a bead-based quantification system. So our product of choice is the Bangs beads, and they're good because you can use exactly the same antibodies you use to stain the cells as you do in your bead. And we've worked up a couple of tricks that let us go below the level of which the Bangs beads would say. They normally go down to about, I think it's 150 receptors, but we've got it now so we can go down to about 10 and measure that kind of really low level. So what was the other thing you asked? Oh, about the stability of expression. Yeah. Because the auxin it doesn't impact the immune cells, we can leave it in there, but it keeps going up. If we take it out, it depends on the target and the system you've used, but normally we see it maintained for 48 hours, and then you start to see a drop-off at that point there. The assays that we run usually last 48 to 72 hours for the cytolysis as well.
And have you published any of this?
No, but okay, I'll take that away and do it. Thank you.
Just following up on the question, the Claudin 978, you compared that with the CD19 engager, right? Is that Claudin 978? What is the secret sauce that makes it so sensitive?
You'd have to speak to Cullinan.
Is it a bispecific antibody?
Yeah. Yep.
CD19 engager.
CD3. Yeah. They have published this with this molecule.
They have.
Data is out in the public domain, but there's nothing so obvious as to what's so special about it. So we haven't shared that fully.
Yeah. Okay. Thank you.
Thanks. Nice to see the range of assays you have. Very comprehensive. Patients aren't healthy donors. So what's your feeling for in terms of getting enough healthy donors to at least hopefully represent what's happening in patients? And and those sort of questions. Is it feasible, possible to get patient sample, PBMCs, for example?
So I'm not quite sure if I heard you properly, but if I've heard you correctly, are you asking, can we look at patient material and do predictive type?
Yeah, in your assay.
And this is actually something that we would really love to do. Yeah, that would be our main goal.
All right, any last questions? Give him one last round of applause.